Good morning, everyone. Today I'll be quickly talking about a lab project that I did around 2 weeks ago about expression of the thyroid peroxidase, which is or TPO, pretty much a protein and a eukaryotic system and detection of the enzymatic activity. So mainly we just deal with molecular cloning and yeast expression. For this presentation, I'll probably focus most on the methods. Yeah, let's just quickly go over it. So for introduction, yeah, I'm really sorry for the blurry image. I don't think it's copyable. So, TPO is generally a crucial enzyme found in the thyroid follicular cell in human. It plays a central role in synthesizing the thyroid hormone T3, T4, and also does it by oxidizing iodide and catalyzing the iodination and coupling tyrosine residuals on the thyroid globulin. Uh, and yeah, pretty much this is a really important protein that we need to deal with our daily metabolism and everything around that. Yeah, so now going through our objectives. So our project is pretty much going through like obtaining a recombinant plasmid containing a human TPO gene and using a molecular cloning techniques and to construct a eukaryotic expression plasmid and introduce it to pretty much a host, and then for the protein expression, and then extract and purify the expressed TPO protein from E cells, and finally to verify the enzymatic activity using the thyroid, like the peroxidase specific assays. Cool. Now I'm going to move on to like the method part. So first of all is to obtain the TPO gene. Yeah, so first of all we use this wait, what is the app called again? Yeah, first we just went online and found the human TPO coding sequence, and then we just downloaded down and then use Snapgene to add in the main primers as well as the homologous boxes or recombinants. Uh, yeah, and then we send it to the company so that they can make the actual DNA for us so that we can keep going with our like later research. Then it's onto the PCR system. I didn't really take a photo of that, but this is a list, I guess, a chart for what we did with the PCR. And the program. So we used the mix, H2O, the template DNA, the two primers. We pretty much just throw everything inside and then just throw it into a PCR, and then it just copies all the DNAs for it so that we can get like up to millions of copies at the end of the 2 hour. Then this is gel electrophoresis and DNA purification. So after the amplification, we ran the 1 degree agarose gel to ensure that what we did actually worked. And yeah, as you can see, the target bands are clearly shown and are at expected size. So that means it works. And then we purify it by pretty much filter it down back to the original form and yeah, keep it as one of the main things we use, I guess. Cool. Yeah, and here's the double digestion and vector purification. So after that, we worked with the vector. So we digested our expression vector with two restriction enzymes. In this case, we used BombAge and KPN1. And after the 1 hour incubation at the 37 degrees, angioelectrophoresis confirmed a successful digestion. I'm not gonna read everything down there, I guess, because that's really a lot. And yeah, we excised the linear vector and purified it. On the right, you can see the electrophoresis result. The first two are somewhat similar and is slightly lower than the control group, which means that yes, it worked. And yeah, this is working on the homologous combination. So so now we just plug everything together, right? The DNA and then the vector that we found. So with both the DNA, the TPO inserted and linearized vector ready, we just conducted the homologous recombination based on the overlap region that we designed in the first step. And yeah, we just did the reaction and it was incubated at 50 degrees, which allows all the protein and the enzymes to work. And yeah, this method just eliminates the need for a traditional ligase and it's way faster, so I decided to use that part. Yeah. So now it's transformation into the DH5 alpha competent cells. So after we added everything together, we throw the DNA into like E. coli, and yeah, we just put it in the ice and then slowly recover that, and then we put it into a medium so that it grows, and then after that, and this is pretty much it. Yeah. Lastly, we took a good like the colonies, I guess a single colony was taken and was suspended in a stir of water. So we used that as a mix and then we pretty much just double check if it worked with the DNA and with a PCR and then we ran the electrophoresis again. As you can see on the right side, the markings are shown and that means yeah, it worked. So yeah, just quickly as a closing, I guess now we need to work on the transformation into the East for expression, but I didn't really have time for it, so I just didn't do that. And yeah, we don't really have much left though. Actually, we all we need to do is just to check whether the enzyme actually works. I mean, well, since we already have the DNA like in our hand, we just need to go check with the company whether all the coding sequences are aligned and whether it's possible to put it into another enzyme. And yeah. I mean, under East actually. So yeah, this is my pretty much my entire presentation. And yeah, please reach out if there are any pro questions or things like that, I guess. Thanks.